Okay, everyone, can you see my screen? Can you see my screen?
Can see, Doctor.
All right. Okay. So can we let's start. So Assalamualaikum warahmatullahi wabarakatuh. Very good morning, everyone. So my name is Dr. Warosalina Waruski. Usually my students call me Dr. Lisa. So you're welcome to call me that also. So I'm from Faculty of Pharmacy. I usually teach pharmaceutical biotechnology there. So for today, I would like to share with you mostly on DNA, RNA, DNA replication, and also DNA damage and repair. So the notes and the questions for ICTL, I've uploaded in the Teams section, in the, I think, the files section, so you can access it there. So to share with you the whole slides, there are about 150 slides. So I don't think it's possible to cover within 2 hours. So we just go through the questions for now. And then if you have anything that you would like to have more clarification on, then you're welcome to actually discuss it with me. Is it okay, everyone? Right. I guess that's a yes. Okay. So for the first question, okay, so the most basic structure when we're talking about nucleic acids, it would be your purine and pyrimidine. Okay, so we would have to understand the structure, okay, in order to understand eventually how the DNA helix is actually formed. Okay, so the structural unit for DNA and RNA is the nucleotide, and this is actually consists of the phosphate group, sugar ribose, and the base. So if you go to the lecture notes, you see that, okay, so this is the sugar. I think that's a better picture. Okay. All right, so actually this would be your sugar, okay, and then your phosphate group and the nitrogenous base. So this is the base that would be either purine or pyrimidine. So purine would have 2 rings, okay, and then your pyrimidine would have 1 ring. Okay, so there are differences in terms of obviously because of the difference in rings. Okay, you would have the size would be different. Okay, and then purine would be consisting of adenine and guanine, and your pyrimidine would be consisting of cytosine, thymine, and uracil. Right. So actually, can you share with me if you have the time to actually go through the questions, or what is usually the style that you do your SCTL? Hmm? Anyone? There are like 100 over students here. Can someone give feedback, or is no one here?
Hello, doctor.
Yes.
Usually we discuss the answer. It's like a
Okay.
Q&A question session.
All right.
So you ask a question and then one of us will answer, and then you'll
Right.
rate the answer if it's correct or not, and give the inputs based on the discussion.
Oh, okay. So it's easier if, because I've done this SCTL with you guys, with your seniors before, but then because it's a face-to-face, then it's easier because, you know, then I can ask you and have the answer sheet with me. Right. So maybe we'll do like that. Okay, thank you for that.
You're welcome, Doctor.
Okay. So, sekejap mana? Okay. All right. Okay, so because I covered the first and second question, maybe you guys want to try the third one. Watson and Crick propose a structure of DNA known as the double helix. So can anyone describe the double helix structure? Anyone wants to try that?
Doctor,
Yes.
if there's no input, you can call out anyone, so they'll have to answer.
Oh, okay. All right. So how about Kirtana? Are you there? Kirtana?
Yes, doctor.
Yes. Okay. Are you aware where are we in our discussion? I would like for you to attempt question 3A. Okay, this is going to take more than 2 hours. Anyone? Anyone wants to volunteer?
Hello, doctor. Can you
Yeah.
hear me? I was a bit lagging just now.
Okay, so
I'm back. It's okay, but can I answer the question?
can you introduce yourself? I'm not sure.
Oh, sorry. My name is Umar Hilman.
Okay. All right, Umar.
Yes. So double helix structure is a double-stranded alpha helical structure with two strands that are connected by hydrogen bonds. Both of the what do you call it? Both of the helical structures are going anti-parallelly. So meaning one of the strands are going from 3N to 5N and the other is the opposite side.
Yes.
And also the both of the strands they pair together according to the complementary base pairing rule, which is AT and CG or A and U.
All right. How about the
nitrogenous
helix?
base pairing.
The helix.
Oh, the helix. So helix is like the spiral shape, helical shape. It's oh, the structure resembles a twisted ladder.
Yes. All right. And it is actually that there's a handedness in the double helix, which is it is right-handed. Okay. So I think Umar, thank you for your answer. I think that's very comprehensive. Okay.
Thank you, doctor.
All right. So basically, it is all that Umar has mentioned. So we have the phosphate group. Okay. that actually contribute to the polar nature of the DNA. Okay, so because the DNA actually have this phosphate backbone, all right, this, if you can see the phosphate group, okay, this serves like a backbone, and because of this it imparts a negative charge to the whole molecule. So we actually use this property to to differentiate DNA from other molecules. Okay, when you're doing your PCR, or for example when you're doing your COVID test, right? You know that it is using PCR. So basically, if you haven't heard what is PCR, okay, it uses the whole principle that we are going to discuss today, which is DNA replication. But then because before that, okay, we have to understand the structure of the DNA. And because the DNA is negatively charged, it can actually be separated, okay, using this charge, because you know that opposites attract, right? So the negative charge would actually be attracted to a positive charge. So this is something that is additional. So when we apply a positive charge to a field, okay, then the DNA would actually be attracted to it, and it will move towards that polar, okay, to that pole, and then there will be differences in terms of its movement because of its size. Okay, so that's something very technical, but then I just want to highlight that it actually uses the polarity of the molecule to actually do this. And yes, it consists of two strands, okay, paired to each other because of the ATGC, the complementary base pairing. And then one is, and it moves anti-parallel, each strand, one from 5' to 3', another 3' to 5'. Why this prime? You can go to the, okay, this one 5', okay, 5', 3', 5', 3', okay. So that's what it means by 5', 3'. And then, okay. Okay, and then we have the narrow and wide groove. So it's this one. Basically, because of the twisting, it has different sizes of the twist. And this actually would give, yes, this one would give a groove. A groove is like a space, okay, a indentation in the structure that allows this that allows other molecules to actually interact with the shape of the whole DNA. So one thing that is interesting about biological molecules is it is not just dependent on the chemical structure or the chemical entity of the materials, but also the structure. Okay, so because of the minor groove, major groove, sometimes we actually use this to actually, you know, impart therapeutic some therapeutic action, okay, because we know of the structure itself. All right, so okay, and then the hydrogen bonds, okay, and then okay, how about B? This is a simple question. The base pairing is according to Chargaff's rule. Explain the Chargaff's rule. So anyone?
Me, Doctor.
Who? Can you introduce yourself?
I'm Muhammad Safwan.
Okay.
So Chargaff rule stated that the ratio of the pairing should be 1:1. So the A and T and the C and G should be in the ratio of 1:1 of each in each DNA sites. I mean, like if A is 20, then T is also need to be 20. If
All right.
so if G is 18, and the C must be also 18, something like that.
Yes, okay. So thank you Safwan, for your answer. Yes, all right. So basically the number of purines equals the number of pyrimidines. Okay, your A would equal your T, and then your G would equal your C. Because it does actually have this complementary base pairing, it pairs with each other. So because of this, the number would be 1 to 1. All right. And all right. So all right, there is always equality in the quantity. All right. Okay, and then the next one, differentiate between RNA and DNA. Okay, how about just a shout out? Okay, someone. Anyone? How about Nurul Shahira? Are you there?
Yes, doctor, I'm here.
All right.
All right, doctor, I'll try the ones that I know.
Okay.
So RNA has one strand and DNA has double strand. The sugar ribose is RNA is made up of ribonucleic acid and DNA is made up of deoxyribose ribonucleic acid. And the nucleotide, I'm quite not sure. Sorry, doctor. And for the Chargaff rule, I think the total number of DNA, they have pairings, and for RNA, since they are single strand, therefore they do not have any pairings.
Okay.
And for the function,
The location? The location.
sorry, location. Sorry, doctor, for a second.
Oh, it's okay. I think maybe another student can help. Thank you, Shahira.
Thank you, doctor.
Okay.
Right.
Hi, Dr. My name is Akash.
Again,
My name is Akash.
Akash. All right. Okay, can you continue from Shahira? Location.
Yeah, the location for RNA is usually in the nucleolus of the cell. And then once it's differentiated in the nucleus of the cell, and then it comes out, the RNA is transported out of the nucleus, and it is duplicated in the outside of the nucleus. That is where the DNA is outside of the nucleus. So RNA is inside the nucleus and DNA is outside the nucleus.
DNA is outside the nucleus.
Yeah.
Okay, it's okay. Okay, and then after that,
And then types and functions, we have RNA is for
hello, Akash. We can't hear you apart from the location there. If you're answering types and functions, we can't hear you. Okay. Anyone? Akash? Why? There are the perks of technical. Okay, how to? Okay. Anyone? Maybe Akash is having some technical difficulties. It's okay. So maybe we finish discussing this first. All right. Anyone want to actually answer types and functions for RNA and DNA?
Doctor, I'm not quite sure about the types, but I can answer the functions part.
Okay, all right, who is that?
This is Anik.
Okay, Anik.
Okay, so for DNA, DNA replicates and stores genetic information.
Okay.
But for RNA, they are involved in the transfer and expression of genetic information.
Okay. All right.
Uh,
Okay, thank you, Anik.
yeah, you're
You,
welcome.
you don't want to attempt the types?
I know the types for RNA. There's mRNA, tRNA, and rRNA, but DNA, I'm not quite sure.
Yeah, DNA is just one DNA. All right.
All right. Thank you very much.
All right. And then next, synthesis, replication, and transcription. Oh, this is a whole lot here. Anyone wants to answer that? All right, so I think because the answer for this last one is quite comprehensive, we just see the answer, okay? And then, right. Okay, so you can see my desktop, right? So yes, the RNA is consisting of single strand and DNA double strand, okay? So, and then you have your ribose for RNA and deoxyribose for DNA. The nucleotide actually differs in terms of your RNA have uracil, but your DNA have thymine instead. Okay. And then Chargaff rule, RNA does not follow this because there's no complementary base pairing, but DNA does. Alright, so RNA is actually in the nucleus and cytosol, okay, and then DNA is only in the nucleus because of the function. So RNA, you know that it also have messenger RNA, right? It carries information from the genes to the ribosome, whereby they would actually build your proteins. So it is also present in the cytoplasm, okay? Meaning that it is transported out from the nucleus. But then the DNA is not transported anywhere. It's just like a blueprint. So it stays in the nucleus. It's not transported anywhere. So RNA is present in nucleus, okay, your mRNA, and then after that it is transported out to the cytosol, whereby it would actually meet with your tRNA and rRNA. Okay, and then for the synthesis, replication, transcription. So RNA is involved in transcription and translation. It means that maybe the question is not really clear. So it is actually involvement in the synthesis, replication, or transcription. So RNA is involved in transcription and translation, particularly in the synthesis of protein. And DNA is involved in replication and also transcription. It is not involved in translation. Right. So the whole story of this is actually very, very interesting, right? Because okay, it's like this. So we have it's in your DNA here, okay, that actually stores all the information. Okay. So from there, your mRNA will be synthesized and then it would be transported out into the cytoplasm, and then it would actually be involved in translation that would involve your tRNA and also rRNA, right? Okay, so the next one.
Doctor,
Yes.
I have a question regarding the structure. Is there actually a significant reason or significant effect as to why the A and the B form of the DNA is like right-handed and the Z form is left-handed? Can you elaborate the different, significant features maybe? Thank you, Doctor.
Okay, thank you. All right, so this one. You're asking why there is a handedness, right? Why do we need this handedness, right? Is it?
Yes, Doctor. Yep.
All right. So usually the one that is present in the the cell is actually B form. Okay, this is the form that exists in cell, and then we have the A and Z form that it is in other other situations. Okay, so you remember that I mentioned that biology is also dependent on the structure. Okay, the 3D dimensions of things. Okay, so the way that it coils, the way that it actually would twist, okay, the direction of the helix would actually impact its shape. You can see that A form, B form, and Z form has different shape, although it is all helix. Okay, the direction would be different. The sizes of the grooves, the minor grooves, the major grooves would be different. Okay, so the one that is present in the cells is B form because of the function that is associated to it. All right, so the function is it is involved in replication and also transcription, right? So all of these processes would also involve proteins. Okay, the histones, okay, we have other polymerases. These are all proteins that would actually interact with your DNA to produce your mRNA. So the whole point, okay, so these proteins are also very specific in terms of its shape. Okay, if you've gone through the topics of proteins, or if you haven't gone through, the whole function of protein is only going to be present when it is folded in a certain way. When it is not able to be folded in a certain way, it loses its function. Okay, so because of this, we know that the folding is very important, and therefore the shape, the 3D shape, is very important. Why it is important? Because it has to interact with your DNA and other proteins. So the one that is present in the cell, which is B form, it is like that because it would be suitable to be to actually do its own to actually interact with other proteins. Okay, and then also it would be actually be able to be packed very very tightly within the cell to actually form your chromosomes. Okay, and at the same time, although it's packed, okay, very densely, it still can actually exert its function at the appropriate time. Okay, so the point is the structure itself is very critical because it has to interact with other components in the whole biological process. Okay, so if you actually differ, if you actually adjust, okay, these structures, it would not actually work as efficiently. Okay, so do you get the answer? Or am I answering your question? Is that from Umar, I think, the question?
Yes, Doctor, it's me, Umar. Yes, you answered my question. Thank you, Doctor.
Okay, all right. So, okay, so can we proceed for the next question? State whether these statements are true or false. Just say TF, whatever. All right, and then I will see whether it is correct or not. Anyone wants to actually join in the discussion?
Hello, doctor.
Yes, hello.
My name is Hafiz. I'd like to try.
All right, okay.
Okay, so for A, it's true.
Okay.
B is false. C is false. D is true. And E is true.
Right. Okay, then we'll see after this. Thank you, Hafiz, right?
Okay, yep.
All right, so the next question, B, anyone? How about Hannah? Hannah Hasri. Are you there? Or Nur Arina? How about Yikin? Yes, all right. Okay, Yikin, can you help us with 5B?
5B, question A is true.
What? Again? True?
Question A is true.
Okay.
Question B is false. Question C is false. Question
Alright.
D is false. Question E is true.
So did I get your answer correctly? Nice. Right.
Yes, doctor.
Okay, so we see. Alright, so TF, okay, because it is actually phosphodiester bond, okay, and then it is not parallel, it's actually anti-parallel to each other. Okay, and then for B, chromatin is actually less condensed and it is actually present in the cytoplasm. So your prokaryotes does not actually have chromatin. If you see these prokaryotes, okay, under microscope, you will see their DNA is just free floating. They don't have nucleus. That's why they are prokaryotes. Okay, and then okay, so it's actually DNA and histone. So the nucleosome is DNA plus histone outermer, and then the linker DNA is actually the DNA between the nucleosome. It does not actually wrap around the histones. Alright, so yeah, everyone got it. Thank you. So the next one. Okay, so you're going to focus on RNA now. Okay, so differentiate between mRNA, tRNA, and rRNA. Anyone?
Doctor, I am Atika. Can I try this question?
Okay, alright, proceed.
All right. So for mRNA, firstly, the structure is like a single-stranded linear molecules. At the 5' end, they have the cap, and then followed by the coding sequence, which is the information that they are carrying, and then followed by the poly A tail at the 3' end. And the function of this mRNA is to basically it's messenger RNA. So the function is to carry information from the genes to ribosome, where it is translated into protein. And then for
Okay, Atika, maybe we can get another person. Thank you for your answer.
okay,
Okay,
thank you, doctor.
alright. So anyone? What about Malvika? Are you there? Okay, you're not there maybe. So Janessa? Ali Akria?
Yes, okay.
Alright.
So,
tRNA.
okay, for the RNA, it's actually a transfer RNA, which are small RNA molecules with a cloverleaf structure. And are the smallest of all RNAs. Internal base pairing forms stem loops, and single-strand molecule have 74 to 95 nucleotide residues. For the function, is to transfer amino acid from cytoplasm and to the protein synthesizing machinery.
All
I
right.
think that's
Okay, so thank you, Ali. You want to add
okay.
to that?
No. Thank you, Doctor.
Okay, welcome. Okay, anyone wants to answer for rRNA? Oh, Malvika is there. Can you take rRNA? Oh, your mic isn't working. It's okay. So, okay. How about Abdul Kadir? Are you there? How about Hari Haran? Are you there? You know, if you open your camera, it would be much simpler for me to actually know whether you're on or not. How about Ruben? Are you there?
Yes, Doctor, I'm here.
All right. Okay. All right. So how about rRNA?
Uh, so the function for rRNA is that it makes up ribosome to the protein.
What?
The function of rRNA is that it makes up ribosome along the proteins, along
Okay.
with proteins.
All right.
And for structure, I don't really get the answer, but it forms three-dimensional in appearance and spherical in shape.
Okay. All right. Okay. Thank you, Ruben. So we're going to actually go through. Okay. Thank you. So the answer. Okay. All right. Okay. So mRNA is a single. Okay, so mRNA is a single-stranded molecule, okay, that would actually have your cap, polytail, and the mRNA structure, okay, and then it would carry information from the gene to the ribosome, and then for tRNA, okay, so tRNA is another molecule that actually depends on its shape, okay, in order to actually exert its function, because this is the one that would actually recognize the sequence in the mRNA, okay, because it has an anticodon. Okay, the anticodon here, okay. If you look at the two-dimensional picture of the tRNA, you see that at the bottom there it has an anticodon. This anticodon would actually be complementary to the sequences in the mRNA. And because of this, it would actually carry the appropriate amino acid. Okay, so you remember that the mRNA would actually tell the whole cell how to build a particular protein. Okay, so it has certain sequences of amino acid that would actually give the protein. So how to do this is it has to know first what is the amino acid required. So tRNA would actually be attached to a certain amino acid, okay, that correspond to the anticodon. So what is what is actually attached to it would be the one required by the mRNA to build that particular protein. Okay, so it would actually transfer the amino acid from the cytoplasm because this amino acid will be free floating. Okay, but then it would actually be taken by the tRNA and then be synthesized into a protein. Okay, so you know that the proteins would consist of your amino acid chain, and then it would be folded, right? So your amino acid chain is built up according to mRNA, right? And then the rRNA, okay, for this one, it has two subunits, right? The structure, it has a 60S subunit and then a 40S subunit. Okay, between this, it would actually help in the translation of the mRNA. If you look at this picture here, so this would be your ribosome, right? Your rRNA. You have your big subunit here, 60S, and then 40S subunit, and then you have the mRNA strand that would actually be put together by the help of the tRNA. The tRNA is not shown in this picture. Okay, but now the structure is it has a big subunit, your 60S, and then the small subunit, which is 40S. So it is actually both of this. Okay, the whole both subunits will actually make up your ribosome, right? So for the next question.
Assalamualaikum, Doctor.
Salam. Who is that?
Hannah. I'm sorry, my internet wasn't good, so I didn't hear you calling me the first time.
Oh, it's okay. All right. So can you help us with the next question? The function.
Yeah.
Okay.
So the cap is located at the 5' end, and it has three functions, which is to protect the RNA from degradation. Then it helps in recognition of mRNA during translation. And then the third one is to stabilize the mRNA.
All
Whereas
right.
okay. Should I answer the poly
Yeah,
A?
you can. Yeah.
Okay. So the poly A is located at the opposite end, the 3' end. So it protects the mRNA from exonuclease attack and to facilitate the translation.
Okay, thank you, Hannah. Okay.
Thank you, Doctor.
All right. So, okay, that is the function. Okay, sorry. The point of why we have the cap and the tail. Okay, so you know that the virus, we can take the RNA virus. Okay, it has the RNA as the genetic material of the virus. So because of this, the cell would actually recognize any foreign mRNA or any RNA strand as something that would be potentially dangerous to the cell. That's why we have all these these nucleases that would actually recognize this floating nucleic acid and then degrade them. Okay, but then how will it actually differentiate between a good, a legit mRNA that comes from the body itself and something that is invading, right? So because of this, the structure of the mRNA, it is actually not tagged as something that, oh, this is ours. No, it is not tagged like that, but it has been given the structure of the cap and tail to actually help in preserving the molecule, okay, from the attack of nucleases. So even though these nucleases would actually, for example, you take your poly A tail there, it has multiple A's. If you actually degrade these A's, it would not actually come into the coding sequences. So when the machinery of your ribosome, mRNA come together to build the protein, it still can have a good working protein. Although the poly A tail is actually degraded by the exonuclease. Okay, and also your cap. Okay, because you will look after this that these nucleases have certain properties whereby it would either degrade from 5' or degrade from the 3' end. Okay, so your 5' cap would actually protect from certain nucleases, and then poly A tail would actually protect from other nucleases. Okay, and then apart from other functions like stabilizing the molecule during the translation processes. Okay, so the whole machinery is quite elegant and actually already taken into account these other possibilities that would actually come into contact when the mRNA goes to the cytoplasm. The safe haven would be in the nucleus because there is no exonucleus there. If you have exonucleus in the nucleus, then your chromosome and all that, it would be degraded. So this exonuclease, this nucleases itself would be present in the cytoplasm. So that's why your mRNA, once it goes out from the nucleus into the cytoplasm, it has to have some protection. So this comes in terms of its structure. All right. So that's that. How about the second question? The function of anticodon arm and acceptor arm in tRNA. Anyone?
Doctor, can I answer this question?
Who's that?
My name is Izdiha, but you can call me Iz.
Okay, Iz.
Okay, so the function anticodon arm functions to form 3 complementary base pairs with a codon during translation in the mRNA. Base pairing as in when guanine pairs to cytosine and adenine to thymine. And then for the acceptor arm, I'm not so sure, but does it have to do with amino acid?
Yes.
So it's attached to the hydroxyl group on the adenine nucleotide, I think. I'm not so sure, sorry.
Yeah, I think that's probably the question. It's actually the attachment site of the amino acid.
Okay.
All right, so you're in the right track. Okay.
Yeah,
Alright,
thank you.
so okay, thank you. So yes, okay, so I think that's clear. Basically, your anticodon would actually recognize the codons in the mRNA and then would bring the appropriate amino acid that is attached in the on the attachment site in the tRNA molecule itself. Right. So okay, all of this. So the next one. Okay. All right. Activity 3. How about the parts of chromosome? Anyone? There, I already show you the answer. Come on. How about the parts of chromosome? Okay, how about Rania there? Rania, are you there? How about Suhash? Are you there? Vishalini? Oh, you have two. Vishalini. Vishalini and Vishalini. Anyone? Any of you?
Doctor, can I answer?
Who's that?
My name is Daniel.
Okay, Daniel.
So you want the structure of the chromosome, right?
Yep.
So there is 3 structures. The centromere, which is the central part of the chromosome, and then the short arm structure at the upper part of the chromosome, and then long arm structure on the lower part of the chromosome. And then within this arm, the DNA molecules are compacted together
All right.
by
Okay.
proteins.
Yeah. Thank you, Daniel, for that
Okay.
answer. All right. So okay. So basically, this is just us making sense of the structure. Okay. Why we have a P arm and Q arm. Okay. Usually, it serves like an address to a gene, though. So how do you communicate to others the location of a gene, right? So usually we would say that it is at certain parts of the chromosome, and we would indicate whether it is at chromosome 2 P arm at certain bands and all this. Okay, so now you know that when it says P arm, it is at the short arm upper structure, and then the Q arm would be the long arm that is at the bottom. Okay, so you know now that the gene is situated where. Okay, so basically that's why we have the parts of the chromosome. Okay, so one of it would be centromere. So anyone would like to share with us the function of the centromere?
Hello, doctor. My name is Amira Nazira. Can I try?
Yeah, please.
Um, Centromere is in the central part of the chromosome, which links the sister chromatids during the mitosis, in which the spindle fibers will attach to the centromere via the kinetochore. And it is also used in the segregation during mitosis and meiosis.
All right. Okay. Thank you. Thank you for that answer. Okay. So if you remember during your, I think SPM, we have meiosis and mitosis, right? And sometimes your body, I don't know about your teacher, but my teacher, we have a dance of meiosis and mitosis, right? So usually how does this two sister chromatids, okay, would be separated or segregated during these processes is it uses a mitotic spindle tau. It is a protein that would actually attach to the centromere, okay, and then it would actually take these two strands apart to the opposite ends, okay, so that is basically the function of the centromere, whereby it would actually interact with the kinetochore to actually function during the mitosis and meiosis to segregate these two chromatids. Okay, right. So classification of the chromosome. Anyone?
Assalamualaikum, Doctor.
Hello, who's that?
My name is Altaf. Can I try answering the classification of chromosome?
Yes. Okay, proceed. Altaf.
So, the chromosomes can be classified based on the position of the centromere. So we have 5 types of classification of chromosomes. Number 1 is metacentric. It means that both arms are equal in length, the P arm and Q arm. And then we have the submetacentric. The arms lengths are unequal, means that they have both different lengths of arms for the P arm and Q arm. And then we have the acrocentric, which means if the P arm is so short that it is hard to observe but still present. And then we have the telocentric, it means the chromosome centromere is located at the terminal end. And then we have holocentric, which means the entire length of chromosome acts as the centromere. That is all.
All right, thank you, Altaf. So I think that's quite clear. Okay, so basically how do we classify the chromosome is dependent on the location of the centromere. Okay, so we now know the function of the centromere, and then that's the function, but then the location of it in the chromosome would actually dictate the classification. Okay, all right, so next question. Explain how DNA helix is wound to form nucleosome. and eventually to form the chromosome. Anyone?
I'll try.
Who's that?
I'm Abirami Abi.
Okay, Abi, you can proceed.
The chromosome DNA will wind around the histone octamer to form a nucleosome, and then the DNA between neighboring nucleosome will bind to histone H1. This nucleosome will further coil up into a shape of helical tubular coils. It's also known as solenoid structure.
Okay.
And that's then chromosome is formed.
Until the chromosome, we have multiple levels of packaging actually. All right. So yeah. Okay. So that is some of the process to produce the nucleosome. Okay. But then after that, nucleosome itself is actually would form a loop. Okay. From the nucleosome, it would form the spindles that we have mentioned, okay, and then it would produce the 30 nanometer fiber, and then it would actually loop more tightly, and then eventually it would actually produce the chromosome. Okay, so if you look at it, okay, so I'm not sure, okay, you can check this this fun fact. Okay, so basically if you actually put it from one end to one end, okay, so each of us has enough DNA to go from here to the sun and back more than 300 times, or around the earth equator 2.5 million times. Okay, so but then all of that could fit into the cell. Okay, so how is that humanly possible? It's not humanly possible, it's only divinely designed. So the way that it is packed, the way that these structures have this fine measurements, okay, that would enable it to be packed and packed again, all right, to fit all of that length into one cell. Okay, it has multiple levels, all right. So and then it's not just a matter of packaging it, but it also has to be unpackaged in order for it to be used for the transcription process. Okay. So how about number 5, the role of histones? Is that actually a question? I think that's already the answer. Okay. All right. So and then next one, activity 4. Okay. Anyone?
Can I try, Doctor? My name is Lina.
Okay, Lina.
For the conservative replication, there's one helix made completely of old DNA and one made completely of new DNA. Whereas for the semi-conservative, there's two helixes that both contain one old and one new DNA strand. But for the dispersive DNA replication, there's two new DNA strands, and they both have a patchwork of the original and the old DNA mixed together.
All right. So yeah. So thank you for that, Lina. So all of you can actually correct the one that I uploaded. I think I put the box in the conservative. It's actually semi-conservative. Okay, because of the complementary base pairing. Okay, a new DNA would actually be built on an existing DNA template. All right. So because of this, one strand is an old strand, and another strand is a newly synthesized strand. Okay, so this is what it means by semi-conservative. It conserves only part of the strand. Only one of the strand is conserved. The other one is newly synthesized. This is what it means, right? All right. So, okay, the second question, the events happening in replication forks. Anyone? Anyone wants to try? What happens at the replication fork?
Hi, doctor. Can I try?
Yes.
My name is Tirbashini. So, at the replication fork, it's actually a branch point where DNA replication occurs. So, in a replication bubble, the replication fork may be unidirectional or bidirectional, where the DNA helicase will break the hydrogen bond holding the two strands together.
All right. Okay. So basically this is the start of the DNA replication process. Thank you for your answer. So firstly it has to actually identify the site whereby it would actually start the replication process, and this site is actually known as the origin. Okay, the origin of replication. So this origin of replication is not just anywhere in the genome. Okay, it has to have a specific base, and usually it's very rich in 80 pairs. Why 80 pairs? If you remember, when we discuss between the complementary base pairing, okay, if you notice that the pairings between A and T only has 2 bonds, but then G and C has 3 bonds. Okay, so this means that it is much harder to actually break the bonds between G and C. So these are stronger bonds compared to A and T. So the AT rich regions would be easier to actually open up because it only has 2 bonds to break. Okay, as compared to a GC rich region whereby it's very strongly held together. Okay, so this origin of replication is very rich in AT pairs because it is easier to open up. Okay, and then after it is opened up, okay, then we would have to actually ensure that these 2 strands remain open, because you remember the complementary base pairing, it is automatic, means that when it has A, it would actually bind to T when it is in close proximity. It's something that is not requiring any energy, so it occurs automatically. So you need to stop that from happening. Therefore, you have proteins that actually would bind to these strands, okay, to actually ensure that it does not renew again. So this is the function of the single-stranded binding proteins and also your DNA helicase. DNA helicase would actually, okay, one thing that you can, a tip that you can use, okay, the name of the proteins, okay, would actually give you a indication of what is its function. Like helicase, okay, it would actually force the strands apart. It would actually break apart the helix. Okay, so helicase, the function is to actually unwind the double helix, and because of this unwinding, it has to stay there. It would actually, that would actually be done by your single-stranded binding proteins. And then because you open up this section, okay, when you open up one section, if you can imagine a structure that is twisted, and then you open up one region, it would actually loosen up that region only, but then it would increase pressure in the neighboring regions. Okay, so to actually relieve this stress, we have to have a way to actually decrease the strain. So this is the function of the topoisomerase. Okay, so it would actually break apart a bit, okay, to actually decrease the strain. So this is what happens in the replication forks. Okay, Okay, next question. Describe the initiation of DNA synthesis. Alright, so what happens during DNA synthesis? Okay, anyone? DNA synthesis is also DNA replication. Anyone? Anyone? Okay. How about Lina? Lina dah tadi. How about Joshua? Are you there, Joshua?
Yes, doctor.
Alright, so can you share with us your answer?
I'm actually not sure about this.
Alright. Okay.
Sorry.
So, it's okay. So the first one, the previous question, we actually discussed about what happens at the replication forks, right? What happens when it wants to start the replication. But then the next question is asking how does it start the DNA replication itself. Okay, now you have opened up that region. How do you start the replication process? Okay, so this is the answer. Okay, this would be the major steps involved in the DNA replication. All right. So, but then it actually focuses on the start of the process. Okay. So the start of the process, you can see. Okay. Okay. So one thing about your polymerase. Okay. Polymerase, it polymerizes. It means that it builds. Okay, that enzyme builds up your DNA. But then satu perangai DNA polymerase, it cannot start from new. Okay, it cannot actually start from nothing. Okay, it has to add on only. It can only add on. So because of that, it needs something to add on to. And this comes in the form of a RNA primer. Primer means a start. Okay. So for the polymerase to start the replication process, okay, it has to actually have a strand to add on to. And this is a RNA, a short RNA strand that is actually synthesized by primase. Primase means that it actually synthesizes primers. Primers would be it's like a catalyst lah. It's like a short RNA strand that would start off the replication process. So after after you have the primer that is built by primase, okay, then your DNA polymerase would actually recognize, oh, something that can be added to. Now I can do my job. Okay, so and then it will go to that DNA and then start adding your nucleotides. Okay, it would start adding nucleotides to synthesize your DNA. Right. So so that's the general process. Okay. After that, we cannot have a RNA sequence in the middle of a DNA, right? So because of this, we have a nuclease activity of another polymerase, DNA polymerase 1, that would actually recognize this RNA primer, and then they would actually degrade this. Okay. So after polymerase 3 have long gone, okay, that it actually would proceed to synthesize your DNA. your DNA polymerase 1 would actually degrade this RNA primer. Okay, and then it would fill in with DNA. Okay. Oops. So that is what happens at this stage. Okay. So the primosome is actually your primase plus your helicase. Helicase, if you remember, is the one enzyme that would actually unwind. Okay, when it unwinds, okay, it would actually be added with RNA primer by primase, and then your DNA polymerase 3 will go there and add on to initiate the process. Okay, is that clear, everyone? Okay. Diam tadi setuju, so I take that you get what I explained. All right. So next question, anyone? The process in leading strands and lagging strands.
Doctor, I could do it.
All right, who's that?
My name is Maha.
Okay, Maha.
The two chains of DNA composed, so the DNA helix has anti-parallel strands. So one of the strand will be 5' to 3'. The other strand, B strand, would be 3' to 5'.
Prime. 3', 5'.
How is it? 5', 3, 5' and 3', and the B strand would be 3' to 5'. So during DNA replication, one of the newly synthesized strands are complementary to that of the parent strand. So for the strand A, which would be 5' to 3', the daughter strand would be 3' to 5', complementary, and similar to the B strand. During DNA polymerases, the synthesis of DNA strand can only be in one direction, that is 5' to 3'. So that poses a problem for our strand 1 because it needs complementary daughter strand. For that, the synthesis takes place discontinuously in series of short fragments called DNA short fragments with polarity of 5' to 3' and RNA primers. These short fragments are called Okazaki fragments. For the joining of the fragments, these RNA primers needs to be removed. So the enzyme nucleases is used that creates nicks, which then are joined together by DNA ligase, hence creating a 3' to 5, 3' to 5' lagging strand. For the leading strand, because the complementary strand had to be synthesized in a proper the 5' and 3', it is synthesized. It is called leading strand, hence forming the semi-discontinuous replication.
Okay, Maha. So because you refer to your answer as A and B strand, so it's not quite directly clear, but then the whole idea of it is correct. Okay, so basically one thing is your polymerase, your DNA polymerase, other than it can only add on, it also only goes from 5' to 3' direction. Okay, so because of this, when you open up the DNA, okay, you have two strands that you have to replicate. Okay, you don't only focus on one strand. Okay, so one of them would be 5' to 3', another one 3' to 5'. So the complementary strand that is synthesized would be the opposite of that. Okay, so you would have one strand that the polymerase can actually continuously add on to, because it is working from 5' to 3'. So that is the leading strand. It just would synthesize continuously without any interruptions. Okay, because it follows 5' to 3'. But then the other strand has to also be replicated at the same time, because you want to close up that section. You cannot finish one strand and then wait for the other strand to actually finish up. So because of this, biology, okay, the design of this is it would actually synthesize the opposite strand, which is the lagging strand, in fragments. Okay, so how does this happen is by the formation of Okazaki fragments. Okay, so it has this initiation of the replication done multiple times. Okay, so you have to do a new primer each time that the fragment is started. Okay, so you have your RNA primer, and then your DNA polymerase 3 would actually add on until it comes into contact with another RNA primer. Okay, because now it cannot add on anymore because another primer is there. It takes up the space. So because of that, it would actually synthesize this lagging strand by fragments, and this is the Okazaki fragments. Okay, but the processes would be the same. Okay, whereby after that, after the polymerase 3 has done adding into the primer, that RNA primer would actually be degraded, and then the gap is filled by polymerase 1. Polymerase 1, it has this exonuclease activity that can actually degrade the RNA primer, and then it can actually add on to fill in that gap. That is the function of DNA polymerase 1. And then how to actually connect between these Okazaki fragments, you have another enzyme which is DNA ligase. Okay, ligase is the one who ligates. Ligates is to glue together. Okay, that is the function of this particular enzyme. It would glue between the fragments. So now you have both fragments intact. Okay, so I hope that's clear. Okay, the differences between leading strands and lagging strands would be because of the direction. Okay, 5' to 3'. Okay, 3' to 5' and all this. Just remember that your polymerase only functions 5' to 3'. So that's why your lagging strand has to be synthesized in fragments. Right. Next one, outline the salient features of replication. Differentiate between prokaryotic and eukaryotic replication process. Okay. Alright, so this is what is happening. Okay. So salient features just means the important major features. Okay, so this is what we have gone through just now. Okay, the major steps involved in DNA replication. First, it would identify where to start. And then after it opens up that section, it would actually put in a RNA primer by the primase. Okay, because your polymerase cannot do from nothing. Okay, so it has to add on to the RNA primer. And then the DNA synthesis would actually commence with your polymerase. Okay, and then your RNA primer would be degraded by DNA polymerase and then filled in by DNA polymerase 1 also. And then these gaps, between these fragments would actually be glued together by your DNA ligase. So that would be the major features. So what are the differences between prokaryotic and eukaryotic DNA replication? Okay. So eukaryotic would actually duplicate its entire genome. Okay. Prokaryotic, and then the eukaryotic DNA would also duplicate its entire genome prior to cell division. Basically, both prior to cell division. So for prokaryotic, it is a continuous process. But then for eukaryotic, it only occurs during the S phase. Why it is S phase? S is synthesizing phase. That's why this is the phase whereby you do DNA replication. So for prokaryotes, they don't have any nucleus, so everything takes place in the cytoplasm. For eukaryotes, we have nucleus, so it takes place in there. The DNA of bacteria and other prokaryotes is circular, but ours is linear. It means that it has an end. It is linear. It is not connected together. Okay, both are double-stranded. Okay, and then there's small amount of prokaryotic DNA, much less than eukaryotic DNA. And then, okay, there is no packaging that is formed in the prokaryote DNA, but then we know that for eukaryotes, it is actually packaged into your genome. Okay. For bacteria, for prokaryotes, we have single origin of replication because it's circular and it's small. But then for eukaryotes, it has multiple origins of replication. Okay, it is to actually decrease the time. If you only have one origin of replication, imagine the time it would take to actually finish up the whole replication process. And then there are different polymerases that are actually functioning in both prokaryotes and eukaryotes. You go through the notes, and then both have Okazaki fragments because both are double-stranded. So one would be leading strand, another one lagging strand. So DNA gyrase is required for prokaryotes. This is the same function, more or less like topoisomerase. Okay, and then for prokaryotes, it's a very rapid process because it is very small. It is small genome, and therefore, and the polymerases are much more efficient. Okay, and then the final product for prokaryotes will be 2 circular chromosomes, and then for eukaryotes, it has 2 sister chromatids. Right. All right, and then the next question, okay, the proofreading process. The proofreading process would be basically it's the same as when you proofread your own work. Okay, you would check whether there are any errors or not. Okay, so we know that for the polymerase, okay, they have proofreading activity, means that although they would actually add on, okay, but it can also have the ability to actually check whether the incorporated bases are correct or not. Okay, so and then it also have exonuclease activity, okay, particularly for your DNA polymerase 3, although it's a the major processing enzyme, it also have exonuclease activity, okay, but it only have the ability to actually correct one base. Okay, right, so we're running out of time, so basically we're going to just go through it. So for activity 5, the types of damage to DNA. Okay, so this one. Okay, so we have your single base alteration. It means that, okay, this is something, damage to DNA is done after the DNA replication process. Okay, but there are also some of them is during the replication process, but usually it is actually addressed by the exonuclease and proofreading activity of the polymerase. Okay, but then we do have also those mechanisms. All right. So we have a single base alteration. It means that it changes single bases. Okay. Those are some of the examples. Okay. And then sometimes it happens by two bases. Okay. So this is when you are exposed to different mutagens. Okay, that actually causes your DNA to be damaged. Where is it? Okay, so this is an example of your depurination. Okay, and then your thymine dimers, and then deamination. Basically, the changes to the DNA after the replication process. So you have your DNA, and then it is actually damaged. Okay, it can be exogenous and endogenous things that would actually cause. These are the types of DNA. All right, this is in the notes. So the mechanisms, they have, okay, we have your mismatch repair. They would actually copy, they would actually address the copying errors. Okay, and then your base excision repair. This is due to this is basically macam, it's like if you imagine your DNA, okay, as something that has to be maintained. Okay, it has to be maintained because it is the blueprint of the cell. So when any damage is done to it, okay, it would have mechanism to try to correct it. Okay, if this doesn't happen, then your cell would actually be destroyed, or it would be destroyed. Okay, if your cells are functioning properly. Okay, but before it goes to apoptosis, it would actually try to repair them, and this could use your mismatch repair, your base excision repair, or nucleotide excision repair, or if it is a very significant damage, then it could also be by double strand break repair. So this is a very major mechanism, okay, to repair your DNA. Okay, so in the rare genetic disease, the underlying cause would be the defect in this mechanism. Okay, so although the body has these mechanisms to actually correct whatever may go wrong, okay, so if there are defects in this process, it could lead to diseases, and one of them would be xeroderma pigmentosa, right? And then, all right, so for the last one, okay, this one is just okay, because you will actually go through this in your what? Okay, all right, during the cell cycle and tumor suppressor gene. Okay, so you will go through this more later, but just to highlight that that P53 gene is actually involved in repair. Okay, DNA repair mechanism. Because of this, if the protein is actually damaged, then we have no protection against any defects that may happen during the that may defects of the genes. Okay, if this protein is defected, then this is why usually cancers associated with defects in p53 protein. Okay, so that's a very fast go through the questions. Okay, so any questions? Any questions? Okay, so if you don't have any questions, I think our time is up. Okay, so basically this DNA damage mechanism, okay, want to actually correct any errors, okay, after the replication process. Okay, so this is what I mean. So after the replication process, they will check. So and then if there are any errors, then the mismatch repair system would actually correct those errors. Okay, this is after the replication process, after the proofreading, after the exonucleus activity yang kita dah cakap, discuss before. Alright. So after that, you have risk of actually being exposed to mutagens after the replication process kan. So this they would actually use other mechanisms like your base excision ke, your nucleotide excision ke, or other mechanism. Okay, so basically if this mechanism is defective, it would actually lead to diseases, and particularly if it involves the p53 gene protein, it would actually cause diseases like cancer. Okay, so I think that's it. Do you have any questions?
Doctor, regarding the repair, right? So would you say that right after replication, the newly synthesized DNA is more, or you could say it's mostly vulnerable to mutations? So are you saying that can we say that it's after the replication, it's like the most vulnerable to mutation, something like that, doctor?
Okay, so I would not say that that stage is most vulnerable to mutations actually, because we have mechanism to actually correct the mistakes, right? Because during the replication process, we know that those polymerase have proofreading activity and ability to actually cleave out those errors by exonucleus activity, and then they can fill in those gaps by polymerase 1. Okay, so after that, they would go into another process of actually checking whether that molecule is intact or correct according to the template. So this would actually activate your mismatch repair system. Okay, if comparing the three major stages of the DNA lah kan, if you can see during replication, after replication, and then during normal cell cycle. Okay, basically after the replication process. Replication process, immediately after, and then after the replication process. After the replication process, for example, in skin cells, okay, it would be most exposed to mutagens. For example, your UV light, kan, exposure to sun, okay, overexposure to sun, or if you're dealing with chemicals, it would actually be exposed to those chemicals that may be mutagens, right? Or even environmental pollutants. I would say if comparing these three stages, the most critical in terms of needing protection would be after the replication process. Okay, because now it has exposure to all kinds of mutagens. Okay, and then depending on what kind of mutation that it actually causes, it would activate different mechanisms that the cell has. Okay, so if all of this fails, okay, then only it would actually proceed to killing of that cell, okay, in terms of apoptosis. If something happens in the genes that actually um, stop the process of apoptosis, for example, P53, okay, it could actually lead to cancer, because you know cancer are like mutants in your cells, right? It actually shrouds itself as if it is normal, but it's actually not normal. Okay, so uh, if those mechanisms would actually fail, okay, then you have a cancerous cell. Okay, so if comparing those three stages, I would say that um, immediately after it would not be most vulnerable. I think it most vulnerable after the replication, because now it is open to mutagens now, right? So you can see that P53, after its activation, it would lead to cell cycle arrest, apoptosis of senescence. Senescence is tua, menua, it would aging. Okay, basically the cell will age and die off. Apoptosis, killing of the cell. Cell cycle arrest, it would actually stop any functioning of the cell. Okay, so basically if this particular protein P53 is defective, then you have a cancerous cell. Okay, so I hope I answered your question. I think that's Umar, is it? Okay, so I hope all of you go through the process of replication and the notes. Okay, I like to imagine this whole biological process like a story though. It's like you have a whole army there that would actually be activated if there is anything that goes wrong. Okay, but when it goes wrong, then you have other mechanisms at that at standby. But then if those go wrong also, then it will be taken over by diseases, right? So it's very interesting. So I hope that you go and explore this topic by yourself, okay, in your journey to explore the knowledge. One thing is, because you're doctors, right, you want to actually treat all of these diseases. So you need to know your enemy. In order to know the enemy, you have to know all of this structure, apa dia punya function, dia punya benda macam ni kan. So why? Because then only you would know their weaknesses, right? Okay, that is how we actually deal with the COVID-19. We look at the spike proteins, what would actually it interact with, kan? Where do we actually target to actually break the chains ke whatever, right? So in order for you to break the rules, you have to know them basically. Okay, so I think that's that. Anything more? Any questions?
Doctor, before you end, regarding the attendance, we are required to answer a question from you.
Oh.
To
Yeah,
verify so any question will do. It's either related to the lecture or not. Doesn't really matter.
Oh, okay. So do I have to check the answers?
Um. No, not really. Actually, it's just sort of to confirm that we are actually in the class because you're gonna ask the question. You see, and
Alright.
yeah.
Okay. Okay. Okay, jap. So I have to type it in, right?
Yeah, sure. Any sort of form would actually do.
Okay. Okay, just a simple one. Okay, I think I've given it already. So I would like for you to actually name the enzyme responsible for unwinding the DNA helix, right? Okay, so yeah, the tip is whatever the function, you just add ACE, ASE, that becomes an enzyme, right? You want the enzyme to be, polymerizing things, then you what? Polymerase. It's not really imaginative, but that's how it is, how you name the enzyme, okay? So what is the enzyme responsible for unwinding the DNA helix? Okay, so basically, for example, another example, the enzyme that synthesizes the primer, we name it primase. And the enzyme that actually glues things together, like ligate things together, we name it ligase. Right? So name the enzyme responsible for unwinding the DNA helix. Okay, everyone? So I hope the session has been interesting for everyone. Okay, I hope you gain something from the session.